Students, in this video we are going to discuss a very interesting mechanism which combines our endocrine and paracrine systems, right? We are going to discuss how exactly Sertoli cells and Leydig cells talk to each other and at the same time coordinate with our pituitary gland. So let's try to understand it in a step-by-step manner, right? First of all, we will draw our two glands, Leydig cells and Sertoli cells, right? We are going to represent them with two boxes, right? Leydig cells then we are going to draw a capillary, right? And then we are going to draw our Sertoli cells. Right? Now the whole story is going to start with hypothalamus, the master controller. Right? Our master controller hypothalamus is going to release GnRH. Hypothalamus. What is going to release? It is going to release GnRH, right? And this GnRH is going to act on your anterior pituitary, right? This will act on your pituitary, right? Anterior and posterior. It will act on your anterior pituitary and it is going to release LH, right? This LH is going to act on the Leydig cells, right? We'll draw a receptor for LH, right? This LH will act on the Leydig cells and it is actually a G protein coupled receptor, right? Let's label it Leydig. Okay, let's label it Sertoli. Now, as soon as the LH act on this LH receptor, right? Since we said that it is a G protein coupled receptor, so what it does that it increases the level of adenyl cyclase, right? It is going to increase the level of adenyl cyclase. Right? And then because the adenyl cyclase level has increased, right? So more of ATP will get converted to cyclic AMP, right? So cyclic AMP level is going to rise, right? If cyclic AMP level rises, the level of pKa is going to increase. The level of pKa is going to rise. If the level of pKa increases, it is going to increase the quantity of very important protein over here called star protein, right? Star protein. The quantity of star protein is going to increase and this star protein will start a very important enzymatic chain reaction, that is the synthesis of testosterone. So what this star protein will do, that it is going to activate the cholesterol side chain cleavage enzyme, right? So you will have cholesterol over here, right? It is going to stimulate your cholesterol side chain cleavage enzyme. This cholesterol will enter mitochondria, right? This is your mitochondria, right? And after multiple steps, I'll just cut it short over here. After multiple steps, it will get converted into pregnenolone, right? Pregnenolone, and after that it will get converted ultimately to testosterone, right? It will ultimately get converted to testosterone. this testosterone will reach your capillaries, right? That write in short form, T. This testosterone will reach in capillaries, and due to the high concentration over here, it is going to diffuse inside your Sertoli cells. Now this testosterone has reached the Sertoli cells, right? Over here, it is going to bind your androgen receptors, AR, right? And after binding the androgen receptors, it is going to act on the nucleus, right? It will go and bind to your androgen responsive elements over here. Right? After binding with androgen responsive elements, it is going to stimulate the genes responsible for meiosis and spermatogenesis. So genes responsible for meiosis and spermatogenesis will be stimulated, right? It is going to stimulate the genes. Which genes? Which do spermatogenesis, meiosis, and apart from that, it is also going to increase the synthesis of proteins which maintain your blood testis barrier, right? It's a tight junction. So which proteins will be synthesized more? The occludins, right? So blood testis barrier proteins, BTB proteins, right? Like occludins, right? Claudins, right? Apart from that, it is also going to synthesize anti-apoptotic factors, right? Like BCL2. So the sperms won't go apoptosis, right? When testosterone falls down, the sperms undergo apoptosis. But when testosterone is there, it synthesizes anti-apoptotic factors like BCL2, right? So it prevents the sperms to undergo apoptosis. Right? So all these things the testosterone will do, right? Up till now the story is simple. The LH acted on the LH receptor, right? It stimulated adenyl cyclase. ATP got converted into cyclic AMP. Cyclic AMP further increased pKa level. pKa increased the level of star proteins, right? The star proteins stimulated the cholesterol side chain cleavage enzyme, and through multiple steps it became pregnenolone. After multiple steps it became testosterone. Testosterone came inside the Sertoli cell. It bound to the androgen receptor, get carried all the way to nucleus, bound to androgen responsive elements, stimulated the genes of spermatogenesis, meiosis, blood testis barrier proteins like occludins, claudins, and anti-apoptotic factors like BCL2, right? But apart from LH, the anti-pituitary will synthesize one more hormone, and that hormone we call FSH, right? FSH. The FSH will also bind its receptor over here, right? And this is also a G protein coupled receptor, right? Since it is a G protein coupled receptor, what it is going to do? It is going to increase the level of adenyl cyclase. Right? Since it has increased the level of adenyl cyclase, what we can expect now? More of ATP will get converted to cyclic AMP, right? If more ATP gets converted to cyclic AMP, the level of pKa is going to increase, right? And this is going to stimulate the genes which synthesize your androgen binding protein, ABP, right? So it will synthesize the genes for androgen binding protein. This androgen binding protein will bind the testosterone level, right? This is very crucial protein because testosterone binds with androgen binding protein and it keeps the concentration of testosterone high inside the Sertoli cells. High concentration of testosterone is required for all these things: spermatogenesis, maintenance of blood testis barrier, anti-apoptosis. So this androgen binding protein binds with testosterone, doesn't let it escape, maintains high concentration of testosterone inside the Sertoli cells, right? Apart from this, it is also this FSH receptor. This is also going to increase the quantity of transferrin. Transferrin is required for maturation of sperms, right? One effect is androgen binding protein, other effect is transferrin, right? Transferrin. Right? What transferrin will do? It will help in maturation of sperms, right? Okay. Apart from that, this action of FSH on this nucleus is also going to increase the amount of one very important enzyme, and that enzyme is aromatase, right? It will increase the synthesis of aromatase enzyme. And what this aromatase is going to do, this aromatase is going to convert your testosterone. I have written short form T. This aromatase will convert your testosterone into estradiol, right? And here the story gets interesting, right? Many students kind of get surprised and confused when they listen that estradiol is also synthesized inside testis, right? Yes, estradiol is synthesized inside testis and it plays the role of a negative feedback mechanism. That means it keeps the Leydig cells under check. If more testosterone is getting synthesized, this estradiol will get diffuse into the Leydig cells and try to control the process. It's a kind of negative feedback mechanism, right? So FSH, see how beautiful whole the mechanism is, right? FSH is going to increase the level of aromatase. This testosterone will get converted into estradiol, and this estradiol will diffuse into capillaries, and from capillaries it will diffuse all the way into your Leydig cells, and here it is going to act on the nucleus of Leydig cells, right? After acting on the nucleus of Leydig cells, right, it is going to inhibit all the steps in this. It is going to inhibit your LH receptor, right? It is going to inhibit your cyclic AMP, right? It is going to inhibit the steps of testosterone synthesis, right? So it's going to inhibit all the steps, right? It is going to inhibit all the steps of testosterone synthesis. By inhibiting the multiple steps, what it will do, it is going to reduce the level of testosterone, right? So both Leydig cells and Sertoli cells coordinate with each other in running the whole show, right? One very important factor which is synthesized by Sertoli cells during fetal life is your desert hedgehog factor, right? Desert hedgehog factor. In short form, we write it DHH, right? Right? So this desert hedgehog factor comes and binds to one receptor which we call PTCH1, right? PTCH1, right? And what this desert hedgehog factor do? It helps in differentiation of Leydig cells during fetal life, right? if the receptor is lacking, or if this desert hedgehog factor is not secreted during fetal life, the Leydig cells don't differentiate and they don't secrete testosterone, right? Apart from that, this Sertoli cells also secrete a lot of growth factors, right? Like TGF beta, IGF-1, right? So it secretes all these factors which helps in growth and differentiation of Leydig cells, right? Leydig cells also secrete factors like insulin-like peptide, right? In short form we call it INSL3, right? Insulin-like peptide, which helps in growth and differentiation of Sertoli cells. Right? INSL3, right? Apart from that, your testosterone inside the capillaries is going to inhibit the secretion of LH from anterior pituitary, right? So we can draw a line like that, okay? This is going to inhibit the secretion of LH from anterior pituitary, right? Apart from that, when FSH acts on the nucleus also synthesizes one very important compound called inhibin B, right? What we call it? Inhibin B. And this inhibin B is going to inhibit the secretion of FSH from anterior pituitary, right? Inhibin B, secretion of FSH from anterior pituitary, and also it will impede the action of FSH on the receptor, right? Okay. So actually I discussed all this because I found it rather beautiful. It's a beautiful interaction of so many hormones, both paracrine and endocrine interaction of so many things which we are seeing over here, right? So let's sum it up, right? So basically we have got two cells over here, Sertoli cells and Leydig cells, right? The whole game starts with hypothalamus. Your hypothalamus is going to release GnRH. This GnRH will act on anterior pituitary and it will release two hormones, LH and FSH. The LH is going to act on Leydig cells. This Leydig cell receptor, this LH hormone receptor on the Leydig cells is basically a G protein coupled receptor, right? It is going to increase the quantity of adenyl cyclase, right? It will convert ATP into cyclic AMP. Cyclic AMP will further increase the level of pKa. pKa will increase the level of star proteins. The star proteins will start the process of synthesis of cholesterol. It is going to stimulate your cholesterol side chain cleavage enzyme, right? This cholesterol will get converted after multiple steps, will get converted into pregnenolone. Pregnenolone after multiple steps will get converted into testosterone. This testosterone will diffuse back into the nucleus, right, and bind to your androgen receptor. After going to the nucleus, it will bind to androgen receptor elements, right? And it's going to stimulate all the genes required for spermatogenesis, meiosis, and also for the maintenance of blood testis barrier like occludins and claudins. Apart from that, it is also going to stimulate the anti-apoptotic factors like BCL2, which prevent the spermatozoa from undergoing apoptosis, right? Meanwhile, the FSH is going to act on your FSH receptor. It is also a G protein coupled receptor. So ATP will get converted into, it will increase the level of adenyl cyclase. ATP will get converted into cyclic AMP. Cyclic AMP will get converted. Cyclic AMP will increase the level of protein kinase A, right? It will act on the nucleus, right? And it will increase the synthesis of androgen binding protein. This androgen binding protein has got a very vital role in keeping the testosterone inside the Sertoli cells. It keeps the level of testosterone high. so all these processes can take place. Apart from that, this action will also increase the synthesis of transferrin, which is required for maturation of sperms, right? Apart from that, it will also increase the level of one very important enzyme, aromatase, and this is a beautiful example of negative feedback. This aromatase will convert testosterone into estradiol. This estradiol will again fuse back into the Leydig cells, right? And by acting on the nucleus of Leydig cells, it is going to inhibit, right, your LH receptor, your cyclic AMP, and even the whole process of the synthesis of testosterone. It's a beautiful example of negative feedback mechanism, right? Apart from that, the testosterone is going to inhibit the release of LH from anterior pituitary. The nucleus of Sertoli cells will also synthesize inhibin B, right, which is going to inhibit the release of this FSH from anterior pituitary, right? It will also act at the level of receptor and impede the action of FSH, right? Apart from this, there are many other factors through which Sertoli cells and Leydig cells interact, right? Like this desert hedgehog factor, which is secreted during fetal life by Sertoli cells, and it helps in it helps in differentiation and growth of Leydig cells during fetal life. Very important factor. The Sertoli cells also secrete TGF beta and IGF-1, which helps in growth and differentiation of Leydig cells. Leydig cells also secrete insulin-like peptide INSL3, which helps in growth and differentiation of Sertoli cells. It's a kind of beautiful mechanism. So many things interacting with each other, modulating each other, right? And both these cells help in growth and differentiation and also keep each other in check, right? So hopefully this video has helped you in better understanding of how exactly Leydig cells and Sertoli cells interact with each other. Hopefully you found this video useful. Wish you all the very best. Thank you.